Accu-Tell Rotavirus Cassette for feces samples. Preparation. Specimen pretreatment. Unscrew the sample bottle, use the attached applicator stick attached on the cap to transfer a small piece of stool. Replace the stick in the bottle and tighten securely. Shake tube vigorously until sample dissolves into test fluid and let stand for 1 to 2 minutes. Testing. Remove the test cassette from the sealed foil pouch. Hold the sample bottle upright with the tip pointing away from the test performer. Snap off the tip. Hold the bottle in a vertical position over the sample well of the test card. Deliver 2 drops of diluted stool sample to the sample well. Read the result between 5 to 10 minutes. A strong positive sample may show results earlier. Interpreting the results. Negative result. Only one colored line appears in the control line region, C. No line appears in the test line region, T. The absence of the test result line, T, indicates that no rotavirus has been detected by the assay. Positive result. 2 distinct colored lines appear on the membrane. One colored line forms in the control line region, C, and another apparent colored line appears in the test result line region, T. The shade of any color within the test area should be regarded as positive. Invalid result. The control line, C, fails to appear.